Okay, good morning, everyone. We are here to start to present you some lectures from Professor Chiara Dall'Asta from University of Parma in the context of FunShield4Med European project. And also my colleague Dimitris Milordos will present the project for you. We will start with Chiara. Chiara, the floor is yours. You can welcome the people.
Thank you very much, Pantelis, and thank you everyone who is there now. I can't see you, but I know that there are people around. The audience is quite
Let me
crowded.
give you a glance of the people. Here is the people.
Great.
Say hi to Chiara, everyone.
Oh, let me say, oh, wonderful. Thank you very much. Okay, so let's go back. Yes, thank you. So I'm really happy to be virtually with you. Anyway, I'm supposed this morning to talk about, I have mainly two different topics. One is the identification of mycotoxins, parent, emerging, and modified. And the second one is a little bit about EFSA risk assessment, or let me say the novel parts of the EFSA risk assessment and regulation. So of course, I don't know how much familiar you are with mycotoxin, although I'm sure that you have heard about that, and probably some of you are also working on a routinary basis on that. So I will not really enter into small details that will take a lot of time. I will try to give you an overview of the instruments, tools we have available for the identification, and mainly about the problem in general. So first of all, just to give you a definition, mycotoxins are secondary metabolites produced by pathogenic fungi in crops, both at pre and post harvest under suitable environmental conditions. What is more important from a chemical point of view, so also in terms of analysis, identification, occurrence, is that mycotoxins share a large diversity in terms of chemical structure. They can be really diverse also in terms of hosting crops, so many different crops, and they are also really diverse in terms of toxicological effects. This means that we are let me say, tackling or addressing a very, very complex problem. Most of all, or let me say on the top of that, mycotoxins contamination is basically unavoidable. I mean, it's based on pathogenic infection, it's strongly affected by climate change, so by the environmental condition, so we can't really avoid mycotoxins in our crops. What we can do is to do our best to keep them at the minimum. So try to mitigate the presence, the occurrence of mycotoxins, both in the field and along the processing, the production chain. As I told you, we have several drivers. I like to call them drivers that can really change the presence of mycotoxins in our food, in our crops. One is, as I already told, the climate change. We don't have time to go through this topic this time with this seminar, but surely the effect of the climate on mycotoxin presence, and in particular the climate in the Mediterranean area, is one of the main topics of our project of FunShield4Med. Then we have another big issue, that is the co-occurrence. Unfortunately, mycotoxins may or usually co-occur in the same crop, and therefore this makes the situation even more complicated, even in terms of identification analysis. Then, and this is more on the side of the regulation, we are facing a strong change in the dietary pattern nowadays. So we are changing our diet, even in the Mediterranean area. So sometimes regulations are not really appropriate. So a change in the diet means also gaps in the regulation that must be filled in. And everything then is let me say, under the umbrella of the emerging risk. So, of course, it's very difficult to give the flavor of such complexity in a very short time, but Yeah, working on mycotoxins also means taking into consideration all these points, especially when it comes to analysis. So small terminology session, just because we need to understand what we are saying. We can have regulated mycotoxins, emerging mycotoxins, and modified mycotoxins. Regulated mycotoxins are pretty easy. All those, let me say, well-known mycotoxins that are regulated in our food nowadays in the European Union and worldwide, just to say aflatoxins, ochratoxin, deoxynivalenol, fumonisin, patulin, among the most important. Of course, regulated mycotoxins are really a few compared to all the mycotoxins that are known nowadays. For this reason, besides regulated mycotoxin, we have a large class which is called emerging mycotoxins. It's not a mystery. Who knows me knows that I don't like the term emerging mycotoxins because it's really not really appropriate. I would say most of them are fully emerged. So for most of them, we have toxicological data, we have occurrence data. So they are basically non-regulated mycotoxins for many, many reasons. So let me say that usually those mycotoxins that are not fully regulated are called emerging, okay? And so mainly Alternaria toxins. Now we have our recommendation for an Alternaria, we'll show you later. Some minor trichotoxins, eniatins, bovaricins, moniliformin, citrinine as well. Ergot alkaloids, although ergot alkaloids have been regulated few months ago in cereals, but still there are issues to be fixed in terms of regulation. And then the very last class, the so-called modified mycotoxins. This is quite tricky. For those of you who are not familiar, just think about metabolites, which is probably easier to understand. It's modified mycotoxins usually are strongly linked with a regulated one. So basically, you have a parent compound that is already regulated, which undergo through a process of biotransformation in the plants or during processing and leading to a different structure. This is a modified mycotoxins. And it is accepted, let me say, that modified mycotoxins may share a similar toxicity with the parent compounds, but I will try to come back later. Okay, so that's the definition of modified mycotoxin based on the EFSA opinion. I just want to mention here that mass mycotoxins, which are also well known in the literature, are part of the modified mycotoxins. So we called modified mycotoxin any metabolite produced by plants, fungi, or mammals, starting from a parent compound, a regulated mycotoxins, and then we called mass mycotoxins only those that are produced by plants. So they are, in sensu stricto, they are produced only by plants. Parent compounds, so regulated mycotoxins, also share a very broad toxicological effects. They can share usually chronic effects with a very complicated pathways or very complicated, let me say, range of toxicity. They can be carcinogenic at a different extent. So we have some mycotoxins that are known as, let me say, they have been ascertained as carcinogenic to humans. By the way, aflatoxins are well-known liver hepatic carcinogenic agents. Then we have ochratoxin, fumonisin, sterimatocystins, what are, possible carcinogenic to humans, but let me say that there are evidence for a carcinogenic mechanism in animals. And then we have known carcinogenic mycotoxins, but sharing a wide set of adverse effects towards different endpoints. Also, the scenario in terms of occurrence is quite complicated. This picture, I like this picture a lot because it's a clear textbook picture. Usually when you got mycotoxins from textbook or even in the classes in the university teaching, you show this kind of picture showing that aflatoxins are mainly prevalent in the tropical area, fumonisins are mainly prevalent in the subtropical area, and so on. But honestly, nowadays, due to the climate change, the situation is not longer this one. This is a map coming from the very last Biomin survey. Biomin is a company producing feeding additives and working a lot on mycotoxins. They are responsible, let me say, they are used to issue every year a report reporting about the occurrence of large occurrence of mycotoxins in many commodities over the world. So it's, let me say, probably the most comprehensive occurrence data report coming from a private company every year. So the global distribution of mycotoxin in 2022 is the one reported in the picture. As you can see, we are completely far from the textbook scenario. We have very emerging, let me say, high alert area, but mycotoxins are more or less everywhere. And what is definitely important, I'm trying to find out, sorry, my laser. Okay, I hope you can see. What is really important is the co-contamination. As you can see, more than, let me say, about 60% of the samples analyzed, and there are thousands of samples, come with more than one mycotoxin. So the situation, especially when you start to analyze mycotoxins, that you really need to have a multi-analyte method to be able to catch the complexity of the situation. Okay, so coming back, biological factors. What can affect the presence of mycotoxins in our food daily? We have, of course, environmental factors, so climate damages and competitive fungi that may occur in the environment and in our crops. And then we also have biological factors. We need to consider biological factors very carefully, and in particular, the plant-pathogen interaction. Because the interaction between two living organisms, a plant and its pathogen, so the host and the pathogen, may really modulate the occurrence and co-occurrence of different mycotoxins. Even if we are focused on the analysis, this is something we need to keep in mind, because due to the different modulation of the plant and the pathogen, so due to this different crosstalk, we can end up with different let me say, widely different mycotoxins in our food. So before planning an analysis, we can't just neglect what can happen in the field. We need to have a broad picture to really plan the analysis from the beginning. Then, of course, we have the post-harvest and the storage management, which are also really important, because, of course, if you don't take care of all the parameters at post-harvest that may affect mycotoxin accumulation, then surely the total mycotoxin load may increase over time during the storage. And then, of course, the processing that may act as sometimes even as a mitigation action. What can we do in the lab? Of course, we have to face a very high complexity situation. I like to use this scheme, which requires a little explanation, but I think it's quite important to make our mind, to make clear the situation for non-routinary analysis. When you work in routine, or let me say, when your target is to get regulated mycotoxins in regulated food commodities, you usually work with those that are called expected known. So compounds that you know very well, you may expect in your food, you know the structure, you know the combination of mycotoxin and food, you know more or less how to analyze them. There are a lot of guidelines, a lot of recommendations also coming from the European Commission and the EU reference laboratory networks. So you know more or less what to do. And that's quite, let me say, straightforward. I don't want to say easy because it's not really easy, but it's straightforward. There are protocols. So you know that, for example, aflatoxin M1 may occur in milk. So you know that when you have to analyze milk, you have to take care of aflatoxin M1. But then sometimes, and due to the complexity of the current scenario, you can find some unexpected known compounds. Just to say, it has happened to me as well. I was analyzing many years ago, let me say, it was more than 10 years ago almost. I was analyzing cheese at the time for different reasons, and I found ochratoxin A in grated cheese, hard cheese. And I was really puzzled about this finding because ochratoxin A is known to have no carryover in bovine, in cow milk. So it's not expected really in milk. So why it has to be in cheese? And then trying to find an answer to this problem, I thought that grated cheese in the European Union, especially hard ripened grated cheese, so like, let me say, I don't want to mention it, but let me say like Parmigiano Reggiano, although that's not only a problem of this kind of cheese, but anyway, it's my area in Italy. This kind of cheese can be, according to the European Union, can be grated with containing up to 80% of 18% of crust. So over the crust of your cheese wheel, you can have molds. And molds may produce ochratoxin A, especially penicillia. And penicillia are quite happy to grow on cheese, and they are quite happy to produce ochratoxin A in a very salty environment. So for this reason, when you grate your cheese with crust inside, and the crust is not very clean, you can have ochratoxin A and the final product. So that's more or less the explanation. After 10 years, we are at the end of the story, more or less, because we know now why. But, you know, at that time, that was an unexpected known compound. Very known because it was ochratoxin A, but totally unexpected in that kind of commodity. For this reason, you need to tackle unexpected known, you need methods that are able to detect mycotoxin in a wide and a broad range. Then you can have expected unknown. So the other way around, just to say very simply, modified mycotoxin. We know that modified mycotoxins, especially mass mycotoxin, may occur in cereals due to the biotransformation occurring in plants. But we don't know how the cereals may modify mycotoxin. So just to say, we know that if we have done, we can guess to have modified mycotoxin in our cereal, but there are so many different structures that we really don't know how they are in terms of chemical structure, chemical diversity. And then at the end, the most complex situation, there are for sure mycotoxin we still don't know and we still don't expect. So of course we can't do anything for that, but we still be open to new compounds in our food commodities. How we can tackle all this complexity? For sure, mass spectrometry is the most important technique and the one who can really give you a lot of information. At the very beginning, we were on low resolution MS and target methods. That's surely the most affordable protocol, and for sure is the most sensitive and can ensure you really accurate and reliable data, just to say triple, quadruple, just to be really clear. Yeah, using a triple quadruple can help you in getting accurate data in your routinary analysis for regulated commodities and regulated mycotoxin, and can be also used for, let me say, non-regulated mycotoxins, so emerging mycotoxins, if you have a standard and if you have reference materials. Then when you move from low resolution to high resolution, so a bit more fancy technique, you can really increase your analytical capacity and go through and target mass spectrometry. So really exploring the occurrence of these unexpected known or expected unknown. This is quite nice to do. It's really interesting to screen very, very different food commodities and matrices, very complex matrices, and just to explore which kind of mycotoxin can be there. The big advantage of the high resolution MS, in my opinion, is the possibility to go for retrospective analysis. So to go back to your previous analysis and look for some new compounds. Just to say, at that time, the time of the grated cheese, I had only a triple quad, so no high resolution in my lab. So I was not able at that time to go back to previous analysis and see if OTA was also in other grated cheese I had already analyzed at that time. But now that I can have access to high resolution MS in my lab, we can acquire a large bunch of data coming from many different commodities, and maybe in 6 months or 6 years, go back to the data and see if a new mycotoxin, a new modified forms, I mean, newly discovered, newly elucidated, was already there. And this is super useful if you want to define the scenario and even the changes in the scenario for mycotoxins or any contaminant in traces. Okay, not only LC-based methods, of course, it's a matter of your target. You always have to balance the time, the cost, and the accuracy. So for sure, LC-based methods are the most accurate, but they are also expensive, more or less, and they require quite a lot of time. So rapid methods, more specifically strips and dipsticks, can be a good, or let me say a cheap alternative, especially if you work in an industrial setting and you have to make a lot of analysis, let me say at the delivery point for deciding yes or no at the acceptance of raw material, just to say. And then ELISA, which is always a very good option for regulated mycotoxins in regulated commodities. ELISA can't be really used for, let me say, unknown or unexpected, because it may suffer from matrix effect. So it's not easy to, it's not a confirmatory technique. So it's really difficult to use ELISA for new compounds. But for the well-established one, ELISA is surely a good option. So let me go for liquid chromatography and identification and analysis. I will be really easy, not complicated, but I'm a liquid chromatography mass spectrometry person, so that's for sure my technique. And let me say that when it comes to triple, quadruple, or low resolution MS, that's surely the most important technique, the most widely used technique for expected mycotoxins. So let me say regulated mycotoxins. It's sensitive, accurate, and it may also allow somehow light sample cleanup, especially when the matrix is not so complex. Then when you use a very dirty matrix, let me say compound feed or I don't know, spices, then of course you need to work a lot on the cleanup to get the right sensitivity. But more or less, all the regulation, all the let me say the sensitivity required by the current regulation can be easily reached using triple quadruple. Nowadays, the golden standard for mycotoxin analysis is the multi-toxins triple quad analysis. So not only one mycotoxin per time or one class of mycotoxin per time, but multiple class of mycotoxin in the same run. There are several very, very fancy validated methods in the literature. This is one from Rudi Kriska group in Austria. Probably the golden standard is almost 300 mycotoxins in the same run, and it's validated according to the Sanko procedure. So it can be used for quantitative routinary analysis. Of course, they have a very good instrument, and let me say that it's not easy to transfer such a protocol in the normal routine, because you really have to keep your instrument super clean. So very good under an academic situation, maybe very dedicated one, but quite complex if you want to work on a high throughput base, let me say. The same group recently has moved to 2000 biotoxin in the same run. And I don't want to go into this because I know they are super good in analytics, but let me say that this is not affordable outside an academic setting and just for research. So very nice to go for multi-toxins, but we need to be really realistic. And under a realistic scenario, the best approach, in my view, for multi-toxin LC-MS is to go with a matrix-related view. So I need to analyze cereals. Which kind of mycotoxin may I get in cereals? I will set up my method for those mycotoxins. So 20 mycotoxin, 30 mycotoxin, 10 mycotoxin, not more than this, because otherwise you can get into trouble. Okay. Then, of course, if you want to make something more, if you would like to go for something more, let me say, unexpected, you can move to high resolution. For sure, with a high resolution, you can enter or diving into the unknowns. Especially, you can work using on-target screening. It's what I'm doing basically in my routinary life as a scientist in the lab, so working on target screening. We can, of course, monitor an unlimited number of ions, and we can go for retrospective data analysis. So this is super powerful if we want to have an idea of what is outside the regulated mycotoxins. High resolution MS nowadays start to be used also for routinary analysis, although I would say that if your target is compliance, so really regulation, triple quad is still the golden standard, because high resolution MS can be tricky. You can have, for sure it is accurate, but accuracy doesn't mean sensitivity. So you can have sometimes issues with sensitivity and matrix effect. The calibration cannot be or can be unstable, let me say, over multiple range order of magnitude. So you really need to recalibrate your system quite often and in a small range. So let me say that high resolution MS is more for exploring situation or exploring new analyze a mycotoxin or identify, of course, new mycotoxin than for routinary analysis. I would surely go for a triple quad if I have to run high throughput routinary analysis. So as I told you, high resolution MS may be very useful for the measurement of accurate masses, so for the identification of unknown compounds. That's super useful, just to say, for the identification of mass mycotoxins, so modified mycotoxins. And there were a lot of mass mycotoxins identified over the past decade. Many more will be detected in the future. And for sure, the collection of the full scan spectra made available by the high resolution MS, so the TOF MS or the Orbitrap MS, opens the possibility to reprocess the stored data over time, and even to have a trend of contamination over time, just going back to the old samples already acquired. Okay, so I mentioned quite a lot mass mycotoxin. They are extremely important, especially when it comes to the plant-pathogen interaction, because they may play a crucial role in plant pathogenesis. So just to say, DON is the most important mycotoxin in wheat and barley. So when you go for the analysis of cereals, you can't forget to consider DON into your analytes. But DON is also a virulence factor for the plants. It means that the fungus or the pathogen is used to produce DON in a way to use DON for hijacking the defense system of the plant. Once DON is inside the cereal, it starts to change the defense pathways and then to give room to the pathogens to grow and to multiply. So it's a way to prevail in the environment, in the plant at least. But of course, the plants may react. And among the resistance mechanism put in place by the plants, the formation of mycotoxin conjugate can be a very effective one. So let me say the plants or the cereals, wheat, barley, and even different maize as well, they are able to exert phase 1 and phase 2 metabolism on mycotoxins. So conjugate mycotoxin with more polar compounds and then just store this mycotoxin into the vacuole where they can't disturb the plant. So it's a way to get rid of them, and it's a way to protect themselves as a plant from the pathogen attack. Of course, breeders are strongly interested in resistant lines. So they are strongly interested in cereal lines that are able to modify mycotoxins. For this reason, we as analysts and also as risk assessor or people interested in risk assessment, we need to consider this option for the future. So if the breeders will go on breeding and selecting, having in mind resistance to mycotoxins, especially under a climate change scenario, we could end up in the future with a totally different pattern of mycotoxins in our food. And this is something that we have to consider. That's why I always like to stress the opening to non-regulated mycotoxins of our method of analysis. Because if you only look for regulated mycotoxins, you can really overlook a lot of other compounds that maybe are growing in terms of concentration in your commodities due to practices in the field due to breedings and even due to the climate change. So really important to keep your analytical eyes open. So that's the scheme of the biotransformation in plants. As I told you, mycotoxin may enter the plant as any other pollutant. Then the plant may exert phase 1 and phase 2 metabolism, and then store the mycotoxins, or the, let me say, conjugated compound in the vacuole, which is a kind of trash bin for the plant. There are many polar groups, so not only glucosides. I think you probably know, so who is familiar with mycotoxins is usually also familiar with DON3-glucoside. That is the most important, most well-known so far mass mycotoxins, modified mycotoxins. But surely glucose is not the only one. We can have conjugation with malonyl glucose, with sulfate, with other sugars, with multiple, with oligosaccharides, even with ferulic acids or cinnamoyl acids. So very, very complicated picture in the plant. Just to say, I don't want to go through this, but this comes from one of our experiments. We feed the plant in vitro with zearalenone, and we end up elucidating more than 60 different metabolites coming from zearalenone in the plant. Of course, not all these metabolites are relevant from a regulation point of view, of course, even from a toxicological point of view, but having clear in mind that the plant is able to modify with such a large extent the mycotoxin can really drive us to keep our analytical purpose really broad, to avoid overlooking any new coming compounds. Very briefly about emerging mycotoxins. Of course, as I told you, there are non-regulated mycotoxins. There are a lot of different classes. I've already mentioned eniatins, bovaricins, moniliformins. They are more or less everywhere, especially eniatins and bovaricins. They are more or less everywhere, and we still don't know, as mycotoxin experts, why. So we don't know the role played in the plant or by the fungus. What I can tell you is that last night we ran, I mean, my instrument was running over the weekend, so I was at home, so I decided to check the analysis from home, and I saw that in my peanuts, there were a lot of bovaricin. And from my side, it was totally unexpected. I'm not an expert of bovaricin, and I'm not an expert of peanuts, but finding bovaricin in peanuts was quite puzzling for me. But it was definitely bovaricin. So, I mean, eniatin and bovaricin are more or less everywhere. Nowadays, the most relevant emerging mycotoxins, or let me say those that are really ready for the regulation, are the Alternaria one. Alternaria moulds have been studied for a lot of time. Toxicity of Alternaria compounds is pretty clear. They can exert estrogenic, genotoxic, and endocrine disrupting like effects. Especially some Alternaria toxins are clearly genotoxic. So it's time to go for a regulation. Usually alternariol, methyl ether, and tenoazonic acids are monitored, but there are other compounds that are definitely important. They have been already assessed by EFSA, so the regulation is more or less on the way. This is a very nice plot coming from a paper from a colleague of mine, Professor Doris Marco from the University of Vienna. She worked a lot on Alternaria, and it's a big friend of us. We shared quite a lot of work. And this picture is very nice because it may show you how Alternaria toxins, definitely much more than alternariol, methylator, and tinoazoic acid. You can find many different other compounds, can be found in several commodities, let me say unrelated commodities, tomato sauce, wheat flour, sunflower seed oil. So basically, products from the Mediterranean area, and basically products that we can find in our dishes every day. So it's clearly a sign that under the climate change scenario, Alternaria will be one of the main alerts in the future. Okay, I will show you later in the second seminar. Now we can maybe have a break with some question, I think, that Alternaria recommendation was already issued by the European Commission, but no regulation is in place. It's different to have compounds under a recommendation than having compounds under regulation. Especially, this is different for companies, especially small companies. Of course, mycotoxin analysis, as any other contaminant analysis, is a big cost for a small company budget. And usually they go for those that are really mandatory for law. Having only a recommendation means that sometimes companies, they are not really positive to increase the number of analyses, so they just screen sometimes for this kind of compounds. So we can really lose the perspective, or we can really lose important information in terms of occurrence in our food. So I think that for this kind of emerging mycotoxins, given the toxicological evaluation, it's really time for full regulation. Different for aniatine, bovaricin, and other unregulated compounds, because in that case, the toxicological effects are not well clarified, slightly clarified, and especially they have higher level, so they require higher concentration to exert their effects. Okay, so coming to the end of the first part, when it comes to analysis, and in particular to the identification of mycotoxins, and when it comes to planning our analysis, we don't have to be reactive. We should try to be proactive. So we need to plan carefully in advance what may go on in our commodities. So surely horizon scanning and the careful monitoring of new drivers may help us in planning our analytical method. Providing high quality data is surely important for occurrence, for compliance, and for risk assessment. I will show you in few minutes that testing for compliance is not testing for risk assessment. So we really need to give to be accurate and give accurate data, high quality data for let me say fit for purpose. Screening is surely important, and screening can help us in having an idea of what is going on outside in the field. And then, of course, confirmatory analysis is surely important for the regulated mycotoxins to be accurate in terms of concentration and for compliance decision. And then my invitation is for scanning for unknowns and to keep our analytical eyes very open to be sure that we can really capture in real time changes in our food, especially in those areas that are more affected by the climate change. So that's my, let me say, idea in terms of mycotoxin identification and analysis. Okay.